Okay, good afternoon, everybody. I have the distinct pleasure of introducing Ian Baldwin to you all. I don't think that he needs much introduction. He is almost a household name in the area of plant-plant and plant-insect interaction. Just a brief summary of what he has done, if I want to give the long story, we are going to be here till tomorrow. But he has done his bachelor in Dartmouth, and then he went for 3 years of training in Penn State, followed by his PhD in Cornell, and then immediately as an assistant professor in the University of New York in Buffalo. So postdocs and students, there is a possibility of going from PhD directly to assistant professor. If you want to know how, ask him. In 1996, he became the director of Max Planck Institute in Vienna. I visited his institute about 2 years ago, and ever since I'm green with envy. But I don't show it, just I'm envious. He has just few papers here and there, about 390 of them, and that a citation of 25,000, I don't, is mind-boggling. He has trained 50 PhD students, and I bet he has dyed his hair. He looks too young. And he has established a new field of studies that he calls genome-enabled field biologist, where he has set up his field studies in Utah, and he really wants to understand the function of genes in response to natural cues under field conditions. He's been nominated and inaugurated for a number of national academies, to start with National Academy of Sciences in US, followed by National Academy of Sciences in Germany, which is called Leopoldina. Then he has so many other German institutions and awards that they all are called wissenschaftliche hervorragende, that means powerful scientist. And he just got the Jean Marie Delaware Award, followed by a grant that makes me against envious, is only 3 million euros to work on clock. And with that, please come and tell me about the timing, which is everything, not only in ecology, I think in doing the postdocs and not. Thank you.
Thank you very much, Katie. Thank you. Thank you for that warm introduction. And I just want to say how wonderful it is to be here in Davis. I think Davis has the most amazing biological faculty in any United States university, and it's just a pleasure being here. I might have a problem with timing. I'm going to talk about some unpublished work, and when I haven't published it yet, it's usually sometimes screw up the timing. But if you do need to leave because I have run a few minutes late, please do, so I know how other people's timing is also important. I'm going to talk a little bit about some unpublished stuff from Sanju Kim, who's here, who's a postdoc, was a group leader and postdoc in the department. He's just moved to Korea to start a CRISPR group there. He's doing CRISPR genome editing on our favorite plant, Mosana Tenada. Philippe Young was a PhD student. He was PhD student number 50 of this genome-enabled field biologists from the department. Mariana Stanton is going to be PhD student number 51, whose work I'll talk about at the end, and Danny Kessler is an engineer in the group as well. So what I want to do is just say a few minutes just about the structure of the Institute. This is the Institute that we finally finished building in 2002. The Institute was started in 1996, and we had a provisional one, but this is where we are now. We have a greenhouse that's adapted, designed for plant-insect interactions, and that was sort of the focus of the Institute in the beginning. So it basically brings together a number of different expertises that are listed here. I don't really want to talk about that. I want to talk primarily about a graduate school that we've set up, the International Max Planck Research School, the IMPRS. And the basic idea of that IMPRS is to address a problem with the training of biologists that has happened since the molecular revolution, and that is that ever since humans evolved, we were excellent natural historians. We knew we have brains, we have nervous systems that are designed almost to acquire natural history information so that we could find lunch and so that we wouldn't become lunch. But ever since we became domesticated, we have sort of lost track of that natural history acquisition business, and we memorize dinosaurs or car models and things like that with that particular nervous system that we evolved. And And the tragedy is even deeper when we look at the training of biologists, where in many institutions it's still very much a situation where people who know how to do natural history are balkanized in different departments from people who know how to use molecular tools. And the basic idea of the graduate training program was to take these particular fields that are typically balkanized in many academic environments, fuse them together in such a way that early stage scientists can utilize those things and become this genome-enabled field biologist, as Katie was mentioning. And what I really mean by a genome-enabled field biologist is really somebody no different from Charles Darwin or Alexander von Humboldt, but with a mass spectrometer in one pocket and a microarray in the other pocket, so that you can use the modern tools of manipulation so that you can actually test hypotheses about function and understand how organisms work in the real world. And that's sort of the agenda. We started out, at least in my department, working on this one particular ridiculous plant that not many people had worked on before. The first publication was in 1922, and I think ours was the next one in 1990 or 89 or something like that. It's a Nicotia tenuata, grows around here. It has a very particular germination behavior. It chases fires in ecologic time. It produces a dormant seed which will last upwards of 400, 500 years in the seed bank if it wants to. Responds to smoke factors that we've identified and germinates the following growing season in the post-fire environment. And because it chases fires, it grows in the agricultural niche. And by understanding how it has survived and all the different traits that it has evolved to grow in that particular niche, we're hoping to be able to translate some of these traits back into our agricultural crops that are forced to grow in this particular niche but never actually evolved in that niche, and make them ecologically smarter, requiring less input. So this is an attenuated natural population that grew in a pinyon, pine, juniper area that was burned. I think this was a 2006 burn, and so this is the 2007 population. And it looks like sort of a field. Our field station is really the unique part of our department. It is located at the Brigham Young University field station, which is owned and operated by Brigham Young. It's called the Lytle Ranch Preserve. And so what we do is basically do all the things that you can do best in German science, which is to do things with a lot of precision and very well endowed infrastructure, and be able to have long-term patient funding, the sort of things that the Max Planck Society allows you to do, things that NSF would never fund, like developing transformation systems for these plants. And be able to do all that in Germany. But of course in Germany, there is no way in the world that you could release transgenic plants into native field environments to understand how genes work. And so that's what we do at that field station. I think we're really the only group in the world that uses a natural habitat to do releases of a native plant. And I just wanted to make some comments about what is a common European attitude about this work, and is that we do this in the US because we can get away with doing releases in an environment which is regulatorily much more lax than it is in Germany. And in fact, that is not the case. We have done lots of releases in Germany until it became totally illegal, thanks to Frau Kunans in 2009. And the regulatory environment is significantly more lax in Germany. We can actually do releases on things that we would never have permission from APHIS to do releases on here. The difference is that it is not an organization in Germany that one can have a coherent scientific dialogue with. And that's something we've been able to do with APHIS for the past 14 years of doing these releases, is having a relatively coherent scientific dialogue where we brought in the natural history information, which has allowed us to do certain types of experiments that we weren't allowed to in the beginning when they were first controlling us. So that's where the field station is, down there. If you fly to Vegas on the only commercial flight that goes from Frankfurt to Vegas, it's a Condor Flight. It keeps the Germans who like to gamble happy. It also gives us a very cheap flight to Vegas on a regular basis. And that's what it looks like down in there. This is the 2013 field plot. This is the 2014 field plot. And this is what it looked like last week when I finished plowing it. And that's where we put in lots of lines. And sometimes we plant plants into native populations, doing different types of experiments. This is a pollination experiment where plants had to be covered so that they would not be pollinated either by a daytime pollinator or a nighttime pollinator to look at the roles of various things. And so this is the number of different lines that we planted, the number of different plants of the different lines, so 43. This year we're going to be doing probably only 36 different lines, but about 5,000 plants, and half of them will be grafted so that the transgenic portion is only the root portion and not the scion portion, that grafting that we can do in the field to look at particular roles that roots play in ecological interactions. I just wanted to point out that this is all that we were able to do in Germany. It took almost about the same amount of work in terms of working with a regulatory agency as this amount of work, and this has generated about 390 papers, and this has generated 2. So the amount of effort that it cost in terms of dealing with the regulatory agencies, and also having your work exposed. I mean, you have to put on the internet not only where the plants are, where they're located, and what's the nearest bus stop you can take to destroy them. So what we do is basically use all the different organisms that live in that particular environment, that interact with that particular plant, to do a number of things. First, to use nature as a laboratory to understand gene function. If we want to understand how genes actually are maintained in genomes, we have to understand how they affect Darwinian fitness. And Darwinian fitness doesn't happen in the laboratory, right? So this is something that basically has to be done for a lot of genes that what a lot of Arabidopsis biologists like to say are, redundant genes. Well, in fact, almost everything we knock down doesn't have a phenotype in the greenhouse. So you don't see a phenotype until we put them out. And I think this idea of gene redundancy is really more a reflection of lousy phenotyping than anything else. So that's one element of it. And we also use the species really as a way to look into an ecosystem in a world of global climate change is what I'm trying to get out of my mouth. And it's told us a lot about that as well, and to do this deep phenotype. And I just want to mention about the deep phenotyping part. All of these organisms make their living off this plant. So they are extremely good at phenotyping these plants. And what we basically do is watch these organisms to help us get a phenotype on our plants that we frequently can't see a difference, but the hummingbirds, the bacteria, and the like can do. So we have an Emboasca leafhopper, which is able to spit into the plant and sense whether or not the plant has intact jasminate signaling. And we can simply use these Emboasca leafhoppers as bloodhounds. We collect them off sunflower, release them to a native population, and they'll find the natural mutants for jasminate signaling. And they've been right 7 times of the ones we found. There's a Trichoparus weevil that is a stem-boring weevil, and that mother who lays the eggs on these plants knows how to identify clock mutants that are natural clock mutants, because changing the clock allows some plants to get bigger, healthier stems, which are better for their babies. We have some bacteria that I've identified, mutants that are deficient in ethylene signaling, and help reduce more sulfur for plants that are sulfur deficient, because when you have a deficiency in ethylene signaling, your yang cycle typically runs like crazy, and you have all sorts of sulfur problems associated with that. I'll tell you a little bit more about that predator, Geocorus pellens, and then I'll talk more about some herbivores that are sort of the key players. And at the end of this talk, if I have time, I'm going to tell you about a new story from Mariana Stanton, who's that PhD student, about Negrobugs that attack the seeds on these plants. So the first part of this story is really just a story about what happened when this wonderful molecule here, nicotine, no longer functioned as a defense as the wonderful insecticide as it functions as for most insects. Because anytime, of course, when you make a beautiful toxin, you're going to select for counter-resistance to that toxin very, very quickly. And that's what's happening to BT, of course, as we express more and more crop plants with BT. We select for insects that are resistant to BT, and we have to figure out ways to deal with that, stacking and adding other toxins and the like. Well, I'm going to tell you a story which is going to screen through about 230 papers, just to lay out exactly how complicated the plant has shaped all of its metabolome, its physiology, and its phenology to deal with the attack of this particular herbivore that has become resistant to nicotine, this guy here. This is a Manduca sexta and Manduca quinquemaculata, are probably the organisms on the planet that have the highest ability to tolerate ingested nicotine. And nicotine is an absolutely wonderful anti-herbivore defense because nicotine obviously poisons the acetylcholine junction, right? That's how it's defined. And anything that has a muscle has to use acetylcholine to be able to move that muscle. And so if you are an herbivore and you're going to have a muscle that moves your jaws or moves your body or whatever around that, having nicotine as your defense compound, which if you don't have a nervous system is no problem, is a really wonderful defense. So this plant undoubtedly, when it figured out about 4.3 million years ago, how to make this molecule, it probably had a wonderful time because dinosaurs and a lot of other things were probably not able to eat this plant anymore. And yet, you know, once you select hard enough on any insect that has a faster generation time than you do, particularly if you have a 400-year seed bank longevity, you're going to end up with organisms that are resistant. And Manduca sexa is one of those organisms. It has an LD50 of nicotine that's about 400 times greater than the most hard-boiled Gauloise cigarette smoker amongst human beings. So it's a pretty impressive thing. So that's what I'm going to talk about first. Then I'm going to talk about some clock components of it and how we think that some of these defense signalings actually hijack the clock rather than the clock anticipating defense on these things, and how trichomes play a role in increasing the apparency of insect attackers as a defense. And then the last story will be about these Negrobugs and how they actually increase the apparency of the plant. They turn it around. While the plant is playing all these games to increase the apparency of insects, there's an insect that turns it around and increases the apparency of the plant. So let me quickly go through those 5 layers. And those 5 layers all start with this particular caterpillar chewing away on the leaf. And in the process of chewing, they lay down a little green slime that you can see right there. And that's simply just the oral secretion of the caterpillar. As it's chewing up leaf, it has to masticate it and get into its stomach. And that's all part of a surfactant property of these compounds that are shown here on the side. These are called fatty acid amino acid conjugates. They're simply just fatty acids esterified to an amino acid, glutamate or glutamine, different fatty acids. There are 8 of them, and these are the signal molecules that plant uses to recognize that it's being attacked by that herbivore. And it actually shows a specific response to that herbivore and not Manduca quinquemaculata. This is the sexta component, and not any of the other 11 other Lepidoptera that will occasionally be found feeding on these plants. So it's a very specific recognition response, and as I say, it activates this 5-layered response that includes defense, avoidance, and tolerance that I'm going to scream through right now. But first, let me tell you about the signaling. So here's an FAC. There's an elicitor that functions as an elicitor. It interacts with some membrane-bound receptor. We have not yet identified that. It activates a complex series of kinase signaling which feed into the jasmate cascade that has a number of differences from Arabidopsis, particularly in its jazz components and the number of mix that it activates. There are other elements in this signaling cascade which are also activated by that FAC that involve a CDPK and a lot of mumbo jumbo I'm not going to tell you about, including an ethylene burst that basically, and there's a new LECRK that we've just found. All of this has to do with one very simple fact, and that is that caterpillars do not brush their teeth before they eat a plant. And so when they are chewing on a plant, they're also introducing all these bacterial signals as well. And the plant needs to figure out, hey, is this a bacteria that's attacking me, or is this an herbivore that's attacking me? And I need to activate very different type of defenses, because if you activate all these anti-pathogen defenses, you frequently make yourself more susceptible to the herbivore than you were if you figured out who actually was an herbivore. And so a lot of this other complexity in the signaling has to do with being able to say, caterpillar, not pathogen here. So that's the nature of that. Now, what does all the signaling do? The signaling activates a whole remodeling of the small metabolite metabolome of the plants. It suppresses those nicotine molecules, that the biosynthesis of it. We figured that out. Why? You can ask about me later. It's because the caterpillar actually co-opts the nicotine for its own defense, and it shuts down the nicotine production that would normally upregulate if it was just wounded. But then also it makes a whole bunch of compounds that are not functioning as toxins or poisons, but they are functioning primarily to inhibit that very reason why caterpillars eat plants in the first place, which is to turn plant protein into caterpillar protein and increase, caterpillars' Darwinian fitness. And that involves interfering with the digestion or wearing down the mandibles and the like. So we've developed a whole lot of metabolomic capabilities and transcriptomic capabilities. We're sequencing the genome as well. And the key thing was to figure out ways of, key thing was primarily informatics. And the informatics is really the brainchild of Emmanuel Gegrel and a number of his students, Yotsana Gulati and Deshwang Li. And these folks have been able to put together a series of very sophisticated ways of mapping metabolites and genes together. And the principle is really very simple. We have the elicitor, so we can take a plant and use that elicitor to very precisely, in a kinetic series, elicit a change in transcriptome, a change in metabolome, correlate these together in a self-organizing map, so this is what this is depicting right here, and then develop network analyses that allow us to pin down particular genes that are regulating a pathway, and then when we knock out that particular gene by RNAi or VIGs, we were able to then look at the metabolome again and then nail down in a much smaller and much more focused way the number of molecules we actually have to do the full characterization on. And the full characterization with secondary metabolites usually involves synthesis because you have to confirm the tertiary structure of it. There are many, many more degrees of freedom. Anybody who's tried to do syntheses on small secondary metabolites knows just how much more complicated that chemistry is than it is with typical nucleic acid chemistry or even protein chemistry, where you have many fewer dimensions than you can have. So by being able to combine these together, we've been able to reduce the number of syntheses that we have to do and figure out new structures that the plant is making in response to these spit factors. But we've also learned a lot about plant-mediated RNAi. We've been transforming plants with insect genes so that the plant is producing double-stranded fragments of insect genes, and when the plants are then fed upon by natural herbivores, they silence the genes in the natural herbivores, and then we're able to study the function of those natural herbivores. Well, we figured if human beings invented this, most likely plants have figured out how to do this too. And when you look at the small RNA transcriptome of plants, and this is stuff that was done by Sri Pandey and during his PhD thesis, we see these spit factors eliciting quite large changes in the small RNA transcriptome, and we think that many of these may well be offensive targets. And so that we've developed a whole platform to be able to dissect the small RNA machinery, the RDRs, the dicers, the argonauts that we've silenced, put them together in various combinations so that we can take out different parts of that small RNA transcriptome and then be able to look at the function of them as well. And the procedure is, whether it's metabolites or small RNA transcripts, is the same. We take a plant and take it back out to the field and just plan it out and then ask the ecosystem, what's different about this plant? This plant typically has no phenotype in the glasshouse, but when you put him out in the field, there's usually a very clean and strong answer. So here is a plant where we've taken out one gene that regulates the biosynthesis of a particular group of diterpene glycoside. There are 17 structures there. They are compounds, if you look in the literature, they're supposed to be sweeteners. Well, they turn out to be incredibly effective defenses, even more effective than nicotine is against this caterpillar. And if you take them out, these caterpillars just ravish these plants. This is not real time. This is sped up time. So that's a typical approach that we take to look at the ways in which the plant uses its own metabolites, its own biosynthetic potential to protect itself. Now, that's not the only way that plants defend themselves. They also have another way that's generally called, those are called direct defenses. They also have another way called indirect defenses that involve not directly protecting themselves with their own chemistry, but indirectly by recruiting these guys here, who are all predators that naturally feed on the herbivore. So as any politician will tell you, these are the enemies of your enemy, and the enemy of your enemy, of course, is your friend. And this is the relationship that the plant has developed through another signaling system that involves basically increasing the apparency of a herbivore as it feeds on a leaf to its predators. And that's all through volatile signaling. So this is part of the volatile metabolome that the plants produce. And when those spit factors enter a leaf, as the caterpillar begins to chew on it, the plant produces a Chanel number 5 bouquet, actually 144 different structures, comparable to what Chanel number 5 has in it, although the molecules are different. And there's one particular molecule here called trans-alpha bergamotin, a sesquiterpene, which functions as a long-distance signal, spreads out from the plant, and is released from the entire plant. So there's signals that go out throughout the whole plant, and not just the attack leaf, but the whole plant begins to emit this, and they'll do this for 2 or 3 days after initial spit elicitation. And it attracts this guy here, Geochorus pallens, right here, who's a little generalist predator who's around in the soil, feeds on lots of different things, and it's a good learner. And it basically makes associative learning to be able to associate that smell with the fact that there's a feeding caterpillar on the plant. But Geochorus pallens actually needs a little bit more information because, after all, it's a small insect and the plant is pretty big, and the caterpillar is hidden somewhere in all that green foliage, and it needs a little more, it doesn't have a GPS unit to tell it where the feeding caterpillar is, but the plant provides that information by moving the double bond from that position there to that position there in a common GLV. There are 6 green leafy volatiles that are produced, and by shifting that double bond over, it tells the Geochorus that this is where the caterpillar is, because these are short distance location molecules. They're molecules that really only exist in the headspace of a plant a few centimeters away from a plant, unlike transalpha bergamatine, which is very stable and can go out and diffuse into a much larger area around the plant. So there's short distance components, there's long distance components in the signaling, and it works gorgeously. These predators come up there, slam their beak into the side of the caterpillar, suck them out, and kill many of them. And they're much more effective than many parasitoids that will keep a caterpillar alive much longer than they normally would, because a parasitoid has its own agenda, of course. And so here you can see the tongue equivalent licking away, sucking all the guts out of the caterpillar. Really, be happy you're not an insect. But I wanted to bring up a particular point, and that is that there is an interaction between these direct defenses and these indirect defenses. And here we have Mary Schumann, who is pretending to be Geochorus pallens with a blue poker here. I'm sorry, I'm not getting started. She's basically taking a caterpillar who's feeding on a normal wild-type plant, a wild-type plant that has all its normal direct defenses. It has nicotine, it has protease inhibitors, it has all those good things. And you can see it's a pretty sluggish caterpillar. It's just taking it in the cheek, so to speak. And when she picks it up, it's sort of like a limp rag, Look at this, look at that. Now contrast that behavior with this behavior here, where she's doing the same thing with a caterpillar who has been feeding on, this is a protease inhibitor free plant, but the same behavior happens if you feed him on a plant that has no nicotine in it. And you can see it's a much more pugnacious caterpillar. It's able to thrash around and spit and look at it, beep it away. And that is a nice demonstration about how effective direct and indirect defenses actually work together, because these caterpillars are basically paying an enormous cost of detoxifying those direct defenses. And it's that detoxification cost that basically makes them sluggish and unable to fight back. So you slow down the caterpillar, you make it sort of lethargic and unable to, and the indirect defenses work so much better because the predators can go up and clean them out without any problem. I didn't realize this. I heard a talk about parrots recently where somebody told me there was 2 very interesting things about parrots. It's a whole group of birds that do 2 unique things. One of them is that they feed on immature fruit, which is probably the most defended part of any plant. It's the part of a plant that is most toxic, and they feed on that particular structure on a plant. And the second thing is it's the only group of birds that takes a 4-hour siesta every day. And it's probably exactly the same process here, where they're just kind of hanging out and trying to detox. So, I mean, we get this idea that, vegetables are defenseless and are meant to be eaten and, whatever, and it's because we bred them that way. But, they are toxic things for most things. And those toxins really affect behavior. So the important point is that you are what you eat. There may well be, plants there not only slowing you down, but knocking out genes in your own detoxification system as well, using various mechanisms. All right, did you follow me? That's layer 3. Now we're going to layer 4. Layer 4 is simply the reorganization of the plant's sink-source relationships. These spit factors change the way in which a plant does sink-source relationships. So typically a plant will fix carbon dioxide, make sucrose, and shuffle up to young leaves to be able to grow new leaves again, right? If you do that and you've got a caterpillar feeding on you anywhere, you're just simply making more caterpillar food. And if these spit factors are there, there's a snRT kinase, we've identified that, and that snRT kinase will change the sink-source relationship so that sucrose, instead of going up to young leaves, goes down to the roots, and it bunkers the resource is down in those roots, and it just waits for the caterpillars to go by. And then when the plant is nice and senesced, after the next little rain, we'll get reflowering. And I discovered this by simply going out there in August, and after one of these little monsoon rains, every single plant that was reflowering was a plant that had been defoliated by a caterpillar. And that's how that system works, is that it's simply just building up a resource in the root that it can then remobilize after that particular problem goes by. It's sort of the Mahatma Gandhi approach to defense. You sort of lay low at the right time and move on. That was 4. Okay, 5 is this problem here, and that is that very frequently good guys and bad guys are all wrapped up in the same genome. So this is the bad guy, but that's the mother of that guy there. And so the same genome has a very good guy because this is one of the main pollinators for the plant. And that pollinator was attracted because the plant produced that compound here, benzyl acetone, from its flower that it opens up at night, right as the time when the moths come by. If you knock out the ability of that plant to produce this floral scent benzyl acetone, we did an RNAi on a gene that we had nobody actually identified as being the biosynthetic step for benzyl acetone, but we managed to figure that out by inference and expression levels in the corolla. You can make a plant that's totally scentless, looks perfectly normal. You put them out at night, you put a video camera on it, and you can see that Manduca simply ignores it. Moths very frequently, particularly the night-flying ones, all are oriented about chemistry. They don't care about vision. Vision is not playing any role there. And so if it's not scenting, these moths simply don't pollinate them. And if they don't pollinate, they're not nectaring. If they're not nectaring, they don't oviposit. There's this linkage between oviposition and nectaring that Manduca has, one that Rick Carbon has shown in earlier work as well. And so the plant is able to, by alter that process, is able to alter the number of eggs that's going to get deposited on it, and it does this by doing this. Namely, it produces an entirely new flower when those spit factors are introduced into wounds. And this new flower, the morning open flower, has a completely different phenology. So here's the typical night open flower. N-O-F, night open flower. And it opens at night and begins to scent, and then it does it for 2 nights, and then it senesces. It usually self-pollinates by this time here. This is the time when it's primarily outcrossing here. The morning open flower, on the other hand, stays closed through that first full night and doesn't open until the morning, and it doesn't scent at all that night. And the nice thing about that morning open flower, besides the fact that it's completely FAC elicited, it produced about a week after the caterpillars begin to start chewing on it and introducing those FACs into wounds, and then it just switches into this new type of flower with a completely shifted phenology. The nice thing about these flowers is that instead of being pollinated by Manduca, it's pollinated by these guys here, hummingbirds. And the nice thing about hummingbirds is that they lay hummingbird eggs when they nectar. They don't lay caterpillar eggs. And so it basically solves its Manduca problem by switching sexual systems to another type of flower. So those are the 5 layers that are all mediated by those particular spit factors. Spit factors which, if you had a genome sequence, you would have no idea that this was happening if you hadn't had the ecology along with it to be able to see what the genome was actually, what parts of the genome were being expressed and what phenotypes were coming out when that expression was occurring. Now I want to switch a little bit to this clock project that Katie was mentioning, the brainchild of Sanju Kim and Philip Young. I'm going to talk a little bit about. Attenuata has a clock that's very similar to that beautiful model that the Arabidopsis community has put up. The only difference is it doesn't have a CCA1, so it has this LHY in its morning element and a TOC1. These are all transcriptionally regulated, and ZTL, which is primarily regulated at the protein level. The Arabidopsis model is refined practically every year. It gets more and more complicated, but these are the sort of the core elements in there. And we've gone and transformed plants to be able to knock down these different elements in the clock, and then fished them out. First of all, had to show that they were circadian, and to show that when you put them under continuous light conditions, they continue to show their circadian behavior. Naturally, ZTL does not show circadian behavior at the level of the transcript because it's a level at the protein, but the other 2, LHY and TOC1, show beautiful circadian patterns. And then, of course, we've taken those Attenuata genes and complemented Arabidopsis mutants and expressed them in Arabidopsis to show that they have the classical Arabidopsis phenotypes. And then when you play various games to silence different components of the clock, you can see that you can shift and alter the patterns that I'm not going to talk about in a lot of detail. That'll all be published fairly soon. But as you might expect, if you take out various aspects of a clock, you end up changing parts and behaviors of the clock in different ways. And I just want to bring it back to a floral story here in a moment. So you can do RNAi and all these different things, and they look perfectly normal, except that they have some subtle differences. And the basic idea of this 5-year project is to look at all aspects of the Attenuata life cycle and to figure out what components are actually regulated by the clock, from seed germination right on through to rosette growth, bolting, flowering, and seed set. And I've set up a whole series of seed bank experiments that my great-great-great-grandchildren will hopefully dig out and tell me about how important the clock is and publish that work in the future. So it's a long-term story here. But what I want to do is talk a little bit about the rhythms. The rhythms really are a central part of clock biology. Carl Linnaeus, in 17-something other, planted this beautiful garden, the clock garden, which had different plants opening at different times. And he used that to take his girlfriends out and say, look, the 4 o'clock is open. It must be 4 o'clock. Or the primroses are open. It must be 6, and so forth. And that, really nailed down that the clock was regulating flower opening and scenting and all lots of things. But it turns out nobody's actually looked at it. And I want to show you a little story here that's not yet published about a series of behaviors that this plant has that is all regulated by the clock. And the behavior I particularly want to show you is this waving behavior here. So the flower goes from a down position to an up position at nighttime and scents, and then goes back down again. And it'll do it in a nice circadian way under a day-night condition, but also continuously if you leave it under constant light conditions as well. And it turns out it's a behavior that's all mediated by a clock right here in the pedicel. The pedicel is what drives it completely, and it has a different clock in the pedicel than it does in the leaf or the stem. And this is work done by Lucas Cortez and Yosina Galati. They have done transcriptional analyses of the upper half of the pedicel and the lower half, and it's a differential growth response. It involves auxin gradients, all the sort of typical things you might expect, but it basically involves 3 growth responses during these 3 wavings. Now, what we were interested in, first of all, was the function and also whether or not it was clock controlled. And the first question really is, why do flowers wave at all? And in order to do that, we had to get some very high-tech experimental equipment, something that really you can only afford if you're a Max Planck director. And that meant going down to the McDonald's and getting some soda straws and cutting off soda straws and then freezing the flowers in different positions and then letting moths have access to them. And flowers that we had done basically a vasectomy on. We had antherectomized them, removed their anthers so they could only be cross-pollinated and not be self-pollinated. And then we looked at seed set on these flowers. And this was a time when we had lost Manduka from our field station for about 3 years. So we had to develop a little a tent system that we could let Manduka fly sort of in the big scale that they like to fly around in Germany. And this was a regulatory nightmare to get this allowed because we had transgenic plants in a tent with a moth flying around. And you know how dangerous it could be that a moth could escape and then would have to fly 8,000 miles across the ocean to pollinate the nearest self-compatible plant. This was also making me seriously nervous, and I would have to smoke some cigarettes just to get over it. And, but anyway, here's the data. So you can see that if you have the flowers in this nice upright position like they like to be at the time when the moth is visiting, you get beautiful seed set and seeds per capsule. Once you put it in the horizontal position and once you put it down, the seed set gets lower and lower. So basically, it's a story of dirty dancing. The flower gets it up so the moth can get it in, right? And it happens at a very nicely coordinated time. Let's see if this really works under real world conditions. So here's a movie of it, and I want to be talking about 3 behaviors. I want to talk about the behavior of that waving, which happens in the pedicel, but I also want to talk about the opening and the scenting, because opening and scenting are highly correlated things, and they happen together. And if we just go through our different clock lines, you can see here's what the empty vector does in terms of the movement. Now the interesting thing is LHY is an early waiver. It starts the whole process about 2 hours earlier than the wild types. Talk looks pretty normally, and ZTL is interesting because it's like a Parkinson's patient. It doesn't wave at all. It's just sort of frozen, stays right there, and doesn't move at all. The benzolacetone has another very similar sort of phenotype. That's empty vector. LHY, well, it has a problem with early nocturnal emissions. It starts to emit scent a little bit earlier. Talk is perfectly normal, except it has a little extra evening bump here in terms of its emission rate. And ZTL does not emit at all. It's completely frozen in its emission pattern also. And so when you do these experiments in Germany and you set them up so as the moths have a choice between simply just taking pollen from a wild type plant and pollinating just one line at a time, all of them produce seed pretty well. So when the moth has no other choice, it'll work hard enough to be able to get the nectar in those flowers to be able to pollinate them. And they can set decent seed set and decent capsule number. But if you compete them so that the flowers are competing against each other, so that you're competing an empty vector versus an LHY. Remember, LHY is an early waver and early center. And you can see that by early waving and early scenting, it gets the moth's attention right in the beginning and gets a better seed set. Talk doesn't differ from empty vector, so it's really normal. And the ZTLs do significantly worse than the wild type, probably because even though it's in the right upside position, it's not scenting. And by not scenting, it's simply not on the moth's radar. It doesn't see these flowers. And we've now learned that the scent not only helps for the large-scale orientation of getting the moth to the plant, but actually it helps with the business of getting the proboscis into the flower. If the flower doesn't have the scent component, somehow the proboscis is tasting that scent, and that's helping to get the beak into the flower. It's a very interesting process. Okay, so that was all in the tent. Then we took these guys back out into the field station. This is a 2013 data set I'm going to show you. This is one of the years when we didn't have Manduka moths, so we set them up so that they would get seen only daytime pollinators and only nighttime pollinators. And the way we did that, we put those white Ku Klux Klan hats that I showed you in an earlier picture on them to exclude one or the other. And here is the nighttime data. And remember, these are not Manduka moths that are pollinating. That's the wrong picture. These are little noctuids. And you can see all of them are doing about the same in terms of their seed set. There's really no difference for the different clock lines. Now, for the daytime pollinators, you see that the ZTL, surprisingly, is the only one that's really getting decent pollination. Now, remember ZTL? ZTL has that phenotype. Well, first of all, daytime pollinators are hummingbirds, right? And ZTL does not scent, but it's frozen in this upward position. And hopefully it will have reminded you of this phenotype right here, that morning open flower. And so what we think is going on is that this FAC signaling cascade that's all running through the jasmine signaling cascade, among other things, is hijacking ZTL specifically and generating this temporary Parkinson patient-like phenotype to solve this particular ecological problem. And what Young is right now doing is developing a histag system to be able to tag ZTL so we can pull down elements of the jasmine signaling cascade, and so we can actually work out the mechanisms of all this. But I think this is really the story about a lot of these ecologic interactions. We don't think that the clock is actually helping the plant anticipate herbivores, as some people in the literature say, because herbivores attack at any damn time. And they will attack at any time and shift that around according to lots of things. What we think is going on is that the clock is actually being hijacked by these environmental signals activated by these herbivores and allowing a plant that is normally trying to figure out how to be heterotrophic and how to be autotrophic at certain times to be able to activate the sort of chemistry that it needs to solve ecologic problems regardless of whether or not it's in the phase of being autotrophic or heterotrophic. And so I think that's probably going to be one of the main take-home messages of our clock story. Okay, so that's what I've told you about. And what I want to do now is go to the last story, which is just about this wonderful little Negrobug, which turns the apparency story. No, actually, I'm going to talk a bit about some trichomes first. Plants produce trichomes. Trichomes, this is a trichome here, produces these beautiful little sticky droplets. These sticky droplets have a pretty simple chemistry. These are sugar molecules here, esterified to different fatty acids on the sugar. And those fatty acids, when they are cleaved off and eaten by an insect, they will produce a very particular scent. And that's the story I'll tell you about. Now, if you look in the literature, these acyl sugars are thought to be defenses because when you go out and look at trichomes, you very frequently see insects stuck to them, like Rick Carbon and his students have been finding lots of aphids stuck to these trichomes, and that's thought to be a defense. If you ask a caterpillar, however, when they first hatch out, the first thing they do is they go lick the lollipops off the tops of these little trichomes. They consume those acyl sugars. They actually grow better. They use it as an energy source. It's actually something quite tasty and beneficial for them. But the interesting thing about these ingesting these acyl sugars is that I'm missing a slide here, is that they gives the caterpillar a body odor. And the body odor comes directly from deesterifying those R groups on those sugar molecules, and the caterpillar begins to smell of those little branched chain fatty acids that are esterified to those acyl sugars. And those branched chain fatty acids are the same compounds in baby barf. So if you have a young baby that's been thrown up on your shoulder, that's what you smell like. That's exactly what these caterpillars smell like. They smell like baby barf. And it's a very distinct odor, and it's a very volatile odor, and it's an odor that is also produced when the caterpillars defecate. So here is a movie that shows the caterpillar defecating. It took a long time to make this movie. But the thing about the defecation is that here is a very stinky piece of poop that is falling from a caterpillar's and depositing onto the ground. And that poop will smell for about 6 or 7 minutes because those fatty acids are quite volatile. The ground is very hot. And that poop will provide information for ground foraging lizards and ants. And one of the wonderful things I was able to do with my parents was to have synthetic mixtures of that Chanel number 5 that comes from the caterpillar poop, and simply spray them on sticks, and able to show that ants just love it. They start running up sticks, any stick that you spray with this particular smell on it. And so ants in the neighborhood learn the smell of that on the ground, and they know that even though they're thinking primarily 2-dimensionally, they suddenly start thinking 3-dimensionally and realize there's something up there good to eat, and they run up the plants and eat those caterpillars. So basically what's going on is that these trichomes, by offering up the first meal for the caterpillar as sort of a nice little lollipop, it turns out to be an evil lollipop because it tags the caterpillars for predation, and it does so very effectively. And I have this picture here because my father just died, and I think a lot about him. And my mother and my father are both medieval historians, and they never paid attention to any of the natural history I did, and they came out in 2010 to do these set of experiments with me, and we published a paper together in PNAS on it. And I just wanted to point out that one can have a natural history moment at any time in your life. And for them, it was 85, but I think it's something that we're all capable of doing. Okay, the last story, and I know I'm running over time now, is about how this little beetle turns the tables on the plant. So we all know that plants advertise and increase their apparency when they want to attract pollinators. So they increase their apparency when they're flowering. They also advertise when their seeds are mature and ready for dispersal. So we all know how wonderful a tomato can smell when it ripens, if it isn't one of those commercial tomatoes, and attract seed dispersers who are going to come and ingest it and then disperse the seed somewhere else. Now, if you are a little Negrobug and you make your living off of feeding on seeds only, you're dealing with an extremely ephemeral resource. And this is what the story of this Negrobug is. And I'm going to just very quickly condense it. It's a story in review right now, and hopefully it'll come out soon. Negrobugs cause enormous amount of fitness damage to these plants. They attack the seeds directly. If you bury seeds that have been attacked by Negrobugs, they have much lower fitness than unattacked seeds. Negrobugs feed on 2 host plants, not only the attenuata that I've been telling you about, but also a perennial plant, Nicotiana obtusifolia, that grows much more sporadically and is the less preferred, but is the earlier flowering one. So this one comes out and it flowers in February. This one doesn't start flowering until May or so in our area. And So that's, Attenuata comes into these big populations and burns and disappears for hundreds of years, while obtusifolia is there every single year on the side of the canyon, one or two plants here or there, and is sort of where when these bugs emerge from their overwintering sites, that's where they first go, and then they sit there and wait for an attenuata to come along. And it turns out they behave like good Mormons. They send their males out to find where the attenuatas are. So if you just look at the ratio, the sex ratio on attenuata populations compared to obtusifolia populations, it's a 3-to-1 ratio. It's just like the Max-Planck Society. There's a sort of similar sort of sex skew there going on. Mariana Stenton did this nice experiment where she simply bagged Negrobugs on plants, on attenuata plants, and showed that if you just looked at the arriving Negrobugs, they also came in with a sex ratio of about 3-to-1. So it's primarily males who are out there colonizing those hosts. Now, so that immediately got her to think that maybe there was a volatile signal that the Negrobugs were giving off while they were feeding that was actually the attracting and bringing in the other colonizers. And so she collected the headspace and found a new compound. That compound occurred in tiny, tiny amounts. It's only produced when when those bugs are feeding on attenuata seeds and not on obtusifolia. It's increased, many orders of magnitude in the production, but it's in absolutely tiny amounts. In femtomolar concentrations are released only in the headspace. There's no gland. It's really a hard structure to get, so you have to trap a lot of headspace. This is an enormous amount of work, something you can only really do when you've got a wonderful chemist collaboration down the hallway, Professor Baldwin, who is expert at identifying and synthesizing molecules. So we figured out very quickly it was a 14-carbon aldehyde, but it has these two unsaturations, and figuring out exactly where they were, the stereochemistry of it, and then synthesizing this, of course, is something that takes somebody who has actually thought about these type of abilities. And Professor Baldwin's group did all that work. And then Mariana was able to take it back out to the field station to see if it worked. And sure enough, you put a little bit of this perfume, you could bring the Negrobugs in, and they come and colonize right away. So this is really just how these beetles, they sit there and they find their preferred host. They start to do this emission. They eat on the plant that they like, and they're burping and farting and whatever that is. And this is the pheromone that they produce. And the Negrobugs do significantly better when they feed on attenuata. So it's not only the preferred host, but also the host on which they have the highest fitness on. So why am I telling you this? I'm telling you this because human beings, ever since we were domesticated by our crop plants, are primarily seed feeders. And we don't really think very much about the fact that our crops, which we've made incredibly apparent, are attacked by pests who probably use these sorts of strategies to do it. The Xerovidula, which is one of the biggest pests on soybean, does exactly the same thing as this one does, but nobody's actually looked at aggregation pheromones that are involved in finding the next soybean crop and moving on, and something that I think we can learn a lot from this beetle on. Okay, so it's time for me to close with the usual sort of broader perspective words. We are at a time in our lives when it's actually fascinating to be a biologist because it's become a digital science thanks to cheap sequencing. And we can understand so many things from sequencing lots and lots of different plants and organisms, and learn a lot about their evolutionary history, as we figured out about the evolution of the nicotine biosynthetic pathway and a lot of these FAC elicited responses that we're working on. And there's even this process what's called reverse ecology, right, where you can sequence a whole lot of organisms and then try to figure out that actually there was something interesting ecologically going on. But in the end, if you really want to understand an organism's genome and to make sense of it, you're going to have to go and do that boots in the field work. And we're going to have to be developing training programs for our students that involve things like just doing what Yogi Berra said, which you can observe an awful lot by just watching, and teaching our students to watch and see things, to be able to do that type of phenotyping that is necessary to understand gene function is something that I think we need to be taking much more seriously in our graduate training program. So I have lots of people to thank. Mariana Stenton for that Negrobug story, all the students I get to work with every year in Utah with the field work, the Max-Planck Society, the Brigham Young University, really the only university I think that would allow us to use their nature preserve as a gene expression laboratory. And I just wanted to mention that, of course, if you have a hot story, don't send it to the evil empire. Send it to eLife. And I, of course, want to thank you for your attention.
Any questions?
Just curiosity, how come you came to this exotic plant? Like, what was the trigger to work with this particular Tenada, the Mosana Tenada?
I'm sorry, I was coughing when you said the first part, so I just need to hear that.
The actual reason to kind of step on this plant versus many other species.
Yeah, and I think that I've told you a lot of really interesting stories. I think if there was a Max Planck Society that was willing to fund you on your favorite native plant, you would find a similar level and number of very interesting stories. Everything that lives out there has to have some very interesting tricks to be able to make it from year to year, right? The reason why I'm working on this particular plant is really due to the Shining Path. That was knocking off gringos in the late 80s, and I was doing my thesis work on Nicotiana sylvestris, which grows in Peru, and my wife said, we just had 2 kids at the time, she said, I'm not going to be here if you're going to do field work in Peru when you come back. So I needed to find another plant, and in '89, that's where I moved to Tenada. Yeah.
Do whatever your wife says, okay?
So I was really interested to see that, you went by kind of fast, but it seemed like with the RNAi plants of LHY, you got enhanced seed set with those plants, and you think that's because it's an earlier phase of the flower movement. So why wouldn't that have been selected for?
Yeah, right. And we think we have natural mutants already in LHY which show that behavior. This is a plant that has enormous genetic diversity in a standard population. We found in a single population, we found jasmine mutants, we found clock mutants, we found ethylene signaling mutants, and part of it probably is this coarse-grained genetic architecture that comes from having seed banks that are very long-lived, and when a fire sweeps through, you recruit from this and that and this and age, many different strata. And I suspect that a lot of ecological variation has to do with changing various aspects of the timing of things, and we're going to find probably lots of natural variation in clock lines that control ecologically important traits. I think this is going to be a really common story. Just because, when you're timing something like with a heterotroph that has to do your pollinations for you, you have lots of different heterotrophs with lots of different behaviors, and some of them are going to optimize your fitness better than others.
Yep.
I have 2 questions. The first one is, do you notice any other differences in the phenotypes between the night and the day opening flowers, like in nectar volume or chemistry or anything like that?
Yeah. So we've looked at the nectar volume and the nectar sugar chemistry, and they're identical. What's interesting about the nectar secondary metabolite chemistry is the games that it plays with nicotine. So we've already published a story that you might have read in Plant Journal, where every random X number of flowers on inflorescence has very high nicotine levels, 200 micromolar nicotine levels. And hummingbirds are terrible outcrossers because they just like the trap line. They've got a nest over here. They just want to get as much nectar as possible, so they go flower, 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 flower. They just work their way through. And of course, when you do that, you're just self-pollinating the plant, right? And so how do you get more outcrossing out of a bird that does that? Well, what you do is you randomly poison one, and then the hummingbird comes up to this flower that's full of nicotine, and it goes, ugh, and it spits it out and flies off somewhere else. And we've been able to show that if you knock out nicotine production, and you look at the number of males that sire the seeds in plants that are nicotine-free versus ones that have nicotine, they have many fewer fathers than the ones that are making nicotine. So it really works. So they're playing cool games with nicotine in the morning on flowers, and we're trying to figure out how that works, because everything else about a flower is determinate and highly well controlled, and here you have this random component. And so we're trying to figure out what sort of nicotine transporter and transposable element might be causing that randomness.
My other question question is more social, and I was curious if there's any efforts at the Max Planck Institute to drive your sex ratio closer to 1.
Yeah, we are under,
Go to Germany tomorrow. What are you doing here?
we are under very strict rules to try to change that, and our first retirement is Professor Baldwin, who's an organic chemist. You know how many synthetic organic chemists there are who are females? Almost impossible to do.
Give us some time.
Yeah.
It will change.
Well, there's almost all the females are smart, they're good in industry. Good academia.
I thought he was going to tell me they're either beautiful or educated.
I know how to save my life. I wouldn't say something like that around.
I have a question for you. So you mentioned something about ciscoterpenes, that they go for a longer period of time. So are they lingering or they are constitutively produced by the plant?
They're definitely not constitutively produced. They're only produced after these spit factors are introduced into wounds.
So they linger in the...
And the reason is they don't have double bonds that make them very susceptible to oxidative damage by ozone. So they just stay in the atmosphere longer, and they'll produce the release for the plant. And we have made them constitutively produced. So we have these plants where we put a gene that makes trans-alpha-bergamotine behind the 30-fast promoter. So they scream all the time. They don't scream just when they're attacked. And we have a paper coming out in eLife next week, which you can read all about the consequences of screaming all the time.
So
it's not good.
What happens to the metabolic pathway when you do that?
Okay. Almost no. So it looks like it's pretty much flux control through the terpene sesquiterpene biosynthetic pathway. In other words, you can. There's a.
What is not bottom up? It's top down.
It's top down. Right. So we have a paper in Plant Physiology I can point you to. It's fully characterized.
So you don't change the MET pathway.
We don't see any effect on the MET pathway. Yeah. As far as in terms of the MET pathway products. Yeah. And I just want to say two things. The other way we knocked out the DTGs is that we silence it through the IRGGPS. And you can there's 3 copies of it, and one of them is specialized only for the synthesis of those diterpene glycosides. All the other things that are synthesized through that same pathway are synthesized through the other alleles. And so the carotenoids are fine, the ABA is fine, everything else is... Yeah.
I'm fascinated by the evil lollipops on the tricomes. How common is that chemistry?
Well, many solanaceous plants produce the same group of acyl sugars. They will produce different fatty acids esterified to them, so that Rob Lass has a nice paper showing the great diversity of acyl sugars produced from different tomato ecotypes and wild types that he's collected. We don't have a lot of variation in the esterification patterns in Attenuata. We seem to see pretty consistent ones. They're quite different from the other main solanaceous plant that's out there that Datura feeds on, that Manduca feeds on, is Datura, and Datura has a very different set of acyl sugars with the different acyl, and the ants seem to recognize the difference. So I can spray the Chanel number 5 from Attenuata poop from the Chanel number 5 that comes from Datura poop, and different ant colonies, depending on whether or not they're in an area with Attenuata or Datura, will respond. So there's some learning that goes on in the ants about what scents to pay attention to. And what we haven't done with this story is really shown that it's a plant defense. We haven't shown that plants actually have higher fitness by making it. And we're trying to get at the biosynthesis of that esterification process so that we can make trichomes that only produce sugar without the fatty acids on them, and then see whether or not they actually have reduced herbivore loads.
When you do C11 labeling for photosynthesis for assimilate from the sink to source, do you change the photosynthetic capacity in response to attack, or do you decrease it? I mean, are there any changes, or it's just a source-sink relationship?
That's a very good question. It turns out that these FACs suppress photosynthesis very dramatically in the attacked leaf, but increase it in adjacent leaves. And we think it's through the regulation of RCA, the rubisco activase, which we see changing very strongly in response to those FACs.
Carboxylate in one and do a lot.
Exactly, right. So that you basically build up a whole group of inactive rubisco in the attack leaf, while the active rubisco, the amount of inactive is reduced in the unattacked leaves.
Activase another ATP production, which is required for activase and ATP together to make the carboxylase ready for...
I don't think I can answer that question. I don't know. Yeah. I don't know.
Any other questions? If not, please join me in thanking for this wonderful talk.